Hello, everyone, and welcome to today's webinar titled Bromodomains in HPV Head and Neck Cancer, using NGS to help understand the transcriptional landscape. This webinar is part of the 10th Annual Cancer Research and Immuno-Oncology Week, and I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by TCAN. For more information about TCAN, go to tcan.com. Now let's get started. Before we begin, I want to remind everyone that this event is interactive, and we encourage you to first participate by communicating with other attendees using our new live chat feature during the presentation. You can find that live chat located on the left of your screen. You can also participate submitting as many questions as you want during the presentation. To do so, simply type them into the Ask a Question box and click Submit. In addition to asking questions, we encourage you to participate in 3 poll questions at the end of our speaker's presentation. Click on your answer when the poll questions are displayed. Now, if you have any trouble seeing or hearing this presentation, click on the Help Desk button located at the top of your screen within the navigation bar or from the lobby. And finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits link located in the abstract window below the presentation window and follow the process to obtain those credits. I now present today's speaker, Dr. Gopal Iyer. For a complete biography of our speakers, please visit the presenter tab from the menu on the left of the screen. Dr. Gopal Iyer, welcome. You may now begin your presentation.
Good morning, everybody. My name is Gopal Iyer. I'm an assistant professor at the Department of Human Oncology at the University of Wisconsin-Madison. You can see this beautiful lake over here. It's called Lake Mendota. This is the entire campus located all around the lake. And it's a beautiful time to be here in the summer. I invite all of you over here. And also, if you like to ski and do ice fishing on a frozen lake, you're welcome in the winter as well. So my lab is basically focused on HPV head and neck cancer, and we have another project which studies lung-brain metastasis, and we are asking the question, why do primary lung tumor cells prefer to go to the brain while some of the other cancer cell types do not prefer to establish in the brain and colonize to cause metastasis. So I'm going to talk to you about the biology of HPV infection and head and neck cancer in this talk, followed by the relevance of proteins harboring bromodomains in viral host transcription, and discuss to you some of the findings of the transcriptional regulation of viral host interactions. The work was made possible because there was a beautiful instrument that was introduced by TCAN, which allowed me to actually be very reproducible in terms of generating libraries for sequencing, which can be quite challenging if you are a lab that are involved in making your own libraries. I will talk about this later as we go ahead in the talk. And so I'm interested in bromodomain and exothermal proteins. They harbor bromodomains, and that is involved in various cellular processes, as you can see in the slide. From a basic science perspective, it's involved in embryonic development, cell differentiation, transcriptional regulation, also involved in evolutionary. There's some evolutionary questions about bromodomains. From a clinical perspective or a translational perspective, they're involved also in co-regulating transcription and during viral infection, and also in normal viral transcription as well. It's also involved in not midline carcinoma, and that has essentially spun off a field which developing small molecule inhibitors against these BET proteins, and also trying to understand epigenetic regulation in animal models as well from a therapeutic standpoint. And these inhibitors now have spun off into approximately about 39 clinical trials around the world, simply trying to ask the question, how can we use BET inhibitors to prevent transcription of key oncogenes uh, in various kinds of cancers? So the reason I say cart before the horse is because sometimes some of these BET inhibitors that have been introduced have shown some effects, but mechanistically has not been completely addressed because BET inhibitors cause various a type of inhibition of from standard transcription to non-transcriptional mode as well. So I think sometimes it's better to ask, understand some of the basic questions before to produce the clinical trials. But I do understand why it should be pushed in the clinical as well, because you want to solve or at least find a therapeutic endpoint for some of these cancers that are debilitating. So essentially, the anatomy of the head and neck cancer is primarily where the HPV resides. Here is in the pharynx region, which is essentially called the nasopharynx, oropharynx, and the hypopharynx. So this is where HPV resides, and if you were to take a swab out of this region, you can actually stain for P16, which is a surrogate marker for HPV, which is a completely different story, but that's how it is being done here. And I'm going to focus on HPV16, which is a high-risk HPV subtype in head and neck cancer. And it is predominantly increasing in younger individuals in North America and Europe because of lifestyle changes and so on. And so it is important for us to find ways to dampen the HPV infection. And we will look into that. Some of the ideas that I'll present in this talk might be interesting to pursue worldwide as well. So the biology of HPV16 infected cells essentially starts at the tonsillar crypt. And you can see an expansion as I expanded this one over here. The cells, the HPV infection occurs through this basal layer. Then you have stem cell differentiation over here. And then you have a dysplastic condition followed by carcinoma. And then that carcinoma in situ then gets transformed into an invasive carcinoma. So, but the molecular characteristics are that you have E6, E7 that is contributed by the virus. That then also has some effects on P53 and RB, which we'll talk about, and that really leads to a dysregulated cell cycle through E2F1. And you have a dysplastic condition, primarily which have been identified to TRAP3, F8, and P10. And then there are some mutations that have been identified in PIK3CA and some aberrant expression of SOX2 and TP63 in tumor, which is essentially what causes the cancer here. So the HPV is an 8 kb genome. It resides as an episomal state. It has about 8 genes primarily, and the E6, E7 are critical, known to be oncogenic as well because they control the cellular P53 and RB tumor suppressive genes. However, these episomal genes, genome integrates into the host, and the way it integrates is that typically through the E2 or E1 region, and linearizes and integrates into the HPV cycle and then into to the host and eventually becomes part of the host machinery during cell cycle and so on. So you do have integration occurring in randomly. You could have an integration in enhancer regions. You can have integrations in intergenic regions. You can have integrations in important tumor suppressor genes. But these are all very stochastic, and so that's what makes it very interesting in terms of understanding other integration effects on causing the proliferation of these cancers, of these cancer cells. So, what we are interested in here is that what are these bromodomain containing proteins, BRD2, 3, and 4, and how are they relevant for HPV cancer? So there are about 46 diverse proteins. There are 61 bromodomains, which are structurally clustered into 8 distinct subfamilies. We will just focus on this family 2, which consists of BRD3, BRD2, BRD4, and see how they are relevant in with the head and neck cancer. So primarily, there are BRD4 long isoform, the BRD4 short isoform, the BRD3 and BRD2. The viral protein E2 was co-crystallized at the C-terminus end. A peptide of this was co-crystallized with the C-terminus end of the BRD4 long isoform. BD1 and BD2 are the domains that are involved in co-regulating transcription. There is also, interestingly, the BRD4 short isoform is also known to interact with viral E2 protein of HPV31, which is interesting because it does not have the C-terminal end, but somehow has been identified to interact with HPV31. There is also the A, which has a NLS, and B, which is also involved in hetero and homodimerization as well. And the extradermal domain is also a protein-protein interaction with other known viral viruses like KSHV and MLV and so on. So the functional significance of these bromodomains are that these bromodomain containing proteins can recognize acetylated lysines on histones on nucleosomes and can then co-regulate transcription. And in this particular case, important genes like BCL2, CDK6, and c-MYC are actually are regulated by BRD4 along with other transcriptional components here. However, the claim to fame for inhibiting c-MYC, which is a non-druggable target for a long time, was with the advent of these BET inhibitors, which could then block this BRD4 to bind to these acylated nucleosomes, which were situated on these target genes like c-MYC and CDK and BCL2 and block transcription. And so this was important discovery, and that spun off a whole field of epigenetic inhibitors targeting BRD4 or BET proteins, essentially. So in the context of HPV, BRD4 is essential for tethering E2 to acylated nucleosomes, and this is an episomal state, and then it tethers to this acylated nucleosomes and is known to regulate mitosis as well. So, but however, the multiple interactions that were associated with BRD4 long isoform also led to the question, can we develop nanomolar inhibitors? And indeed, there are some nanomolar inhibitors that have been developed to target the BRD4 long isoform. And as you can see over here, there is the E2 interaction that interacts with BD2 as well, and then here you have E2 that interacts with C-terminus of that. However, most of the inhibitors target the BD1 and BD2 domains of BET proteins. There are some other transcriptional co-regulators and activators that have been identified that interact with BRD4 long isoform, and it is of interest to actually inhibit some of these activity so that you can then downregulate some of the target genes that recruit some of these transcriptional co-regulators and activators. So we looked at this from a context of, can it be used, these inhibitors can be used for regulating some of the viral and host genes in head and neck cancer that were infected with HPV-16. And so what we were interested is in this particular state, the integrated state, where you have high stromal activities, which induces worse survival rates as well. And you can also have low stromal activity, which also creates a proliferative signature. So while, as you can see over here, the episomal state has a better survival rate, while the integrated state has a worse survival rate. So we were mostly interested in the integrated HPV populations. And so for that, we asked some of the questions, because HPV proteins regulate some of the tumor suppressors like p53 and RB as well. And so they sequester it to degradation, and that leads to an aberrant cell cycle. While if you look at the normal physiological pathway, you don't see a lot of changes associated because E2F, p53, and E2F, there's a very nice cell cycle regulation associated with the normal physiological state. While as in the HPV, it is controlled by E6 and E7. So we then curated the HPV TCGA database and simply asked the question, are these cancers, HPV patients, addicted to BET transcriptional activity? And lo and behold, if you look at the transcription TPMs, a lot of these patients do overexpress BRD2, BRD3, and BRD4. That led us to the question, can we use BET inhibitors to target these BRD4 and BRD2 and BRD3 activity as well? So then what we did is we took a panel of cell lines which were HPV infected, which we characterized for integration status, and they were all sensitive to the BET inhibitor JQ1, which is a pan-BET inhibitor, and also you can see that they also sensitize them at low nanomolar to high nanomolar IC50s. However, one of the interesting things was that BRD2 was upregulated, while BRD4 was early effects were downregulated, but then it shows an effect, which is expected because it does not self-regulate itself at this stage. Didn't look like that, but this is more like a protein effects of BRD4 inhibition through JQ1. So then we looked at, from a localization perspective, BRD4 was fairly present all over the place, which is as expected, but surprisingly enough, BRD2 actually went and got sequestered in regions where TAPI was not stained. In TAPI refractile regions, you see a BRD2 located was sequestered into that, and we do not know currently as to why, but we're exploring the idea of why this BRD2 goes and sequesters in this dapirefractile regions. So then we asked the question, if we downregulate SH of BRD4, do they also affect viral genes like E2, E6, and E7? And it was interestingly enough, BRD4 downregulation through SH knockdown also controls E6, E7, and suggesting that it also phenocopies the effect of the small molecule inhibitor JQ1 as well. So essentially, this result basically led us to the idea that we should understand the overall transcriptome of these cells as to what are the differences, and does BRD4 actually control E6, E7, the viral genome as well. And then we went back and we were trying to make a lot of libraries, and the present opportunity presented itself with TCAN, and this is the beautiful TCAN machine, which looks essentially like an extended PCR machine, where you essentially add your RNA sample and magically something happens and it generates you consistent libraries without any hiccups. The machine essentially recognizes these reagent cartridges. It has magnetic beads. It has a lot of sample deck components. It has all the things that you need to do a library prep, including tips and tubes, which you don't necessarily have to do anything. And it is very intuitive, and it eliminates a lot of the sample inconsistencies in the library preps that arises due to, having to pull it off a magnetic rack and making on and off and so on and so forth. So the setup time is essentially, you just load your RNA sample, and if you have DNA, you can do that as well. And you can just walk away from it, and it's a fully integrated system where you can actually just, essentially have everything completely automated, and you can go and have a good time while the library is being made. It has a lot of, it has a reusable frame and stand. The samples goes here in the first row, and it has all the beads and so on. It's already pre-mixed. It has bulk reagents and also has a waste, and it also has tips. So you just load the whole thing over into this cassette, and then you start the machine, and you essentially press this magical button over here, and you walk away, and you come back after 10 hours to get your RNA libraries. And for DNA, I think it's around close to 6 hours. It's pretty neat. It has solved a lot of our challenges that we have had, especially when you're dealing with patient samples, which are very precious. You can do anywhere between 10 to 1,000 nanograms. It does the usual steps that you have located in a manual library preps, but you don't have to pipette. That's what I'm trying to get at. So you just have to walk away with just inputting this RNA, and it will do all these steps of fragmentation, reverse transcriptase, purification, repair, adapter ligation, barcoding, and so on and so forth, and you get a very neat library at the end of it. So in our case, we were very surprised to see that all our triplicates matched consistently, and there was a very good coverage, 5' to 3' transcript coverage, and which is fairly surprising, because when we've done this manually, this was not the case, and this made life very easy, and your total sequencing cost comes down to almost like $50 per sample, as opposed to something that could run into hundreds. We were also surprised that when you compare this to the K562 control samples, they were quite similar in terms of the coverage, and which was very good. And we were fairly happy because all our samples are here and also here, and they were fairly consistent with the control samples as well. If you look at, there was also a very high correlation between the technical replicates when we did the control samples, which led us to believe that this would also be the case for our experimental samples as well. So, so when we also had low library yields, or rather when we had low input, we are also able to get very good library preps out of that as well. So we wanted to see if we loaded a low amount of RNA, we also get similar high libraries as well. So we were very surprised and we were very happy with it. So that reduced a lot of our headaches in terms of doing the manual library prep. So it can also do intact DNA, it can also do fragmented DNA, it can also do mRNA. And so we've done we've done quite a bit of work with this, and so we're fairly consistently surprised as to the quality of what we can get out of this. So then we asked the question, what happens when we do the, compared to the manual RNA, mRNA-seq workflow, you have all these different steps. It takes you about 7 hours, while in the MAGIC NGS mRNA-seq workflow, your sample prep time is only 10 minutes, and then you just walk away, and the libraries are made essentially after 10 hours. But if you feel like doing something else with your life, you can potentially hold your samples up to 65 hours within the machine, and nothing goes awry with the libraries. It frees up time for doing other things as well. So for the DNA-seq as well, this is the same thing. You have 50 to 500 nanograms. It will do all the essential steps that what you do from your manual library prep. And you can work, there are several workflows available which will allow you to do high quality DNA, pre-fragmented DNA, cell-free DNA, and so on and so forth as well. So just to summarize why we chose to run with TCAN and how it made our life easier to understand our HPV biology in terms of transcription and transcriptomes and so on. The success rate was fairly astounding compared to the manual library prep, although you could still get good libraries, but I was more interested to make sure that I could get this in and out time and time again, the same quality, as opposed to having to have some good and some bad libraries. Do you have, typically you don't need any expertise, you just need to load the cassette with the reagents, and then you can just walk away. So yeah, there's a lot of positive aspects of this. I would strongly think that you should test it out simply because it's just reproducible. And for me, and for us in the lab, we would like to produce consistently good libraries so that we are confident of what we are stating and interpreting the data. So then we come back to the biology. So the question here is, while the HPV integrates into the genome, we want to ask a question using these libraries and the sequencing data that we obtained. How do we identify what the HPV reads are? So we adapted something called a discordant read pair, where you can actually then identify where exactly in the genome of the host have they integrated, number one, and then you can also identify from that region how many read counts do you get for your individual viral genes. So you do something which is, a discordant read pair, and you can identify your viral gene and also the host gene, and understand, how whether these genes are co-regulated by BRD4 transcriptionally in the presence of the inhibitor or not. And then we were very surprised to see that when we did something called a nested PCR, that the HPV genome is actually disrupted in the E2 region, while the SCC2 cells were not disrupted in the E2 region; they were full length. And we were able to actually map the read counts, and we identify it was integrated in the X chromosome over here, and here it was integrated in this particular case in chromosome 3. And interestingly enough, it suggested to us that there was some integration-dependent effects on how BRD4 co-regulates transcription, because if you look at the DIAPH2 gene, in both the cases, BRD4 did not seem to regulate DIPH2 gene, co-regulate DIPH2 gene, while BRD4 did co-regulate TP63, and in the presence of the inhibitor, downregulated significantly the host gene as well. So along with the data that I showed you that E2, E6, E7 was also downregulated, we also identified that wherever the insertion did occur in the host, the cellular gene also was downregulated at TP63. So which suggested to us that there was some integration-dependent effects occurring, and BRD4 selectively co-regulates some of the host genes as well in this particular case. So we also looked at all the other the genes that we were interested in. So you could see here that at the protein level, whenever E2 was disrupted, E6, E7 was downregulated, while when E2 was not disrupted, there was no change. So in the E6, E7, which led us to believe that there could be the integration of the viral genome affected how the E6, E7 was regulated by BRD4, and in turn also could be regulated by E2, which is supposed to also have some effects on E6 and E7 activation as described in the literature. So interestingly here, a point to note here is that E6 and E7 gets downregulated significantly, while in this particular case, in this particular line, E2, E6, E7 were not significantly downregulated, but in fact they seem to be upregulated to a certain extent in the presence of, although they are not statistically significant. What is more interesting here is that c-Myc was downregulated at 24 hours here, while c-Myc was not downregulated in the presence of the JQ1 inhibitor as well. So overall, it's suggested to us that E6, when E6 and E7 was downregulated, RB and p53 was also downregulated, while in this particular case, RB and p53 were not significantly affected at 24 hours, as you can see over here. So which suggested to us that there must be something going on uh, in this particular case, and which we're trying to explore at this moment. And you can see from a protein perspective as well that p53 actually comes back, the tumor suppressor activity comes back on, and which is also very interesting. While despite the p53 and RB coming back up in this particular case, in the full length E2, the RB was also coming back up, but had no significant impact towards cell cycle, which I'll show you in the subsequent slides. So in case the summary here is that in truncated E2 cells, BRD4 regulates E6, E7 independently. In full-length E2, BRD4 has no significant impact on E6 and E7 transcription. And on the host side, while whenever there is an disruption, BRD4 is significantly downregulates C2F1 as well. We are independently evaluating this in other lines as well, and it seems to suggest that integration plays a role in how JQ1 interacts with BRD4 in other cases as well. So the questions that we are interested to ask is, why did E6, E7 not alter RB and p53? Why was HPV not impacted by BRD4 regulation in full-length E2 expressing cells? When we looked at the cell cycle, interestingly enough, whenever there was a downregulation of E6, E7, as understandably there was a G1 arrest, significant G1 arrest up to 48 hours. While if you look at the full length, there is a significant G1 which is consistently hovering around 50%, but there doesn't seem to be any cycling of the S and G2 phase as well, so suggesting that there are some other non-RB p53 effects going on. So when we looked at the overall RNA sequencing profile, we recognize that the homologous recombination mismatch repair cell cycle was significantly affected in the case of the truncated E2, while in the full-length E2 there was no significant impact. So it looks like the integration affects the cell cycle and global RNA transcription as well, which we are actively pursuing in terms of understanding how it occurs. So then we asked, can we ask, can we actually use this in the context of radiation? Because currently in the clinic, the standard of care treatment is HPV patient with cisplatin. So then we simply ask the question, how do beta inhibitors directly impact DNA repair genes? And can we use de-escalation of radiation dose with beta inhibition as a potential alternative treatment? So then what we did is we did a standard clonogenic assay where you radiate cells with different doses of radiation, and you make a clonogenic survival assay, and we identified that 6 rays did seem to have an impact as well on the survival of these cells. So then we looked at all these different DNA repair components because typically you cannot have mutually exclusive non-homologous end joining or homologous recombination occurring in a cell, so they're all tied in intimately. But we were very surprised to see that in the presence of JQ1, there was, and radiation, there was a significant downregulation of these MR11, NBS1, and RAD51 as well, which is very surprising because it suggests that they should, in combination with radiation, JQ1 seemed to somehow impact some of these DNA repair genes as well. And so then we asked the question, what should we do in terms of how do we assay this? So we looked at gamma H2X repair formation, and we were surprised that we could still see the gamma H2X foci still being retained at 24 hours, and more of this was retained, suggesting that the repair activity was delayed, which suggests to us that if you can actually add a beta inhibitor plus radiation, you can extend the instability of the genome of these cancer cells, because now you don't have to, these cells cannot repair efficiently, which is a favorable outcome when you want to treat patients. So then we summarize these preliminary results, and then we identified that c-MYC creates cell cycle arrest in G1 and truncated E2 expressing cells. The RNA sequencing profile of truncated E2 and full length were very distinct, and transcriptional DNA repair genes are impacted in truncated E2 cells more significantly than the full length. So we are trying to identify whether if you can combat this with radiation, is this a potential route for creating genomic instability? Because obviously BET inhibition is not sufficient enough to create long-term instability, and you need this radiation as a hammer to exacerbate this instability in these genomes. So currently now we are also exploring new bromodomain specific inhibitors. These are second generations. They have been recently published, and they have had a very interesting take, because you can actually selectively inhibit BD1 and BD2 and simply ask a question, how do they affect target genes as well? So we're exploring this in the context of HPV as well by using the selective BD1 and BD2 inhibitors. So that is what I have to talk about, and I can tell you that a lot of this transcriptional output data that we have had was extensively enabled by using this TCAN machine, which would have not been possible, or rather it gave us more confidence in terms of how libraries were made using that machine. I'd like to thank for your time, and the primary players were this lady, Akarsha Rao, who was in my lab and now has moved on to be a grad student. Albert is a biomedical engineering student, a graduate student in the lab. Sijan did most of the bioinformatic analysis. Hassler was another student who helped in make some of the libraries. Kwong helped in doing most of the protein data. Christina Kendriowski has been instrumental in providing a lot of bioinformatic support in trying to do the discordant read analysis. Randy is a clinician on this project, and Paul Lambert is the godfather of HPV, and he has primarily been very instrumental in supporting most of the work that has been done in my lab. Thank you for your time, and I will take any questions that you have.
Thank you, Dr. Gopal Iyer, for your time today and for your important research. And before we go, I want to thank our audience for joining us today. We would appreciate your participation in answering a few final polling questions. To record your answers, click on the option on the screen. Our first polling question: do you regularly generate libraries for NGS for your laboratory? Yes or no? I will give you a few moments to answer. Our next question: are you considering investing in NGS workflow automation? And finally, would you like to receive further information about Magic Prep NGS sample preparation systems? Thank you so much for participating in those polling questions. I'd also like to take a moment and thank our sponsors, TCAN, for sponsoring today's webinar. Questions submitted today and during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. And this webcast can be viewed on demand for 2 years until October 6, 2024. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Bye-bye.